All right, today we're going to tackle a really powerful tool for anyone who has to deal with massive amounts of data. It's called Principal Component Analysis, or just PCA for short. It's a game changer, so let's jump right in. So, let's set the stage. Imagine you're a scientist and you've just finished a big experiment. You're staring at data for 15 different genes, and you've got that data for 60 different mice. The question is, what do you do now? How do you even begin to find the important patterns in all that information? I mean, if you do the math, that's 900 separate data points. It's a classic case of information overload, right? You can't just compare everything to everything else. You'd be there forever. It would be a total mess. So here's our game plan. First, we'll talk about this too much data problem. Then we'll see how PCA is designed to find the strongest patterns. After that, we'll learn how to actually read a PCA plot, peek under the hood at the math. Don't worry, it'll be painless. And finally, we'll wrap up with why this tool is so incredibly useful. Okay, so let's start with that first big hurdle, the too much data problem. This is a classic saying, you literally can't see the forest for the trees. And in the age of big data, this is a challenge that scientists and analysts face every single day. Well, this is exactly where PCA comes in to save the day. At its heart, PCA is just a method for cutting through all that noise. Its job is to find the really strong, important patterns that are hidden in huge, complicated data sets by, well, summarizing them. And I love this description from one of the sources. It says, PCA takes all that data, all 15 genes for every single mouse, and it just smushes it all down into one single dot, one dot for each mouse. So suddenly you're not trying to analyze 900 data points. You're just looking at 60 dots on a graph. Now that is something we can work with. But wait, how does it do that smooshing? How does the algorithm decide what information is important enough to keep and what it can ignore? Well, that brings us to our next section, how PCA actually finds those patterns. Turns out, PCA is trying to do two things at the exact same time. First, it looks for the direction through all your data points that has the absolute biggest spread, the maximum variation. And second, it tries to create a summary of your data that has the minimum possible error, meaning it loses as little of the original information as possible. And here is the really, really cool part. You'd think those are two different goals, right? But mathematically, they are the exact same thing. Finding the direction with the most variation is the same as finding the summary with the least error. It's this beautiful, elegant bit of geometry that actually goes all the way back to the Pythagorean theorem. A fantastic way to visualize this is to imagine your data points aren't just numbers in a spreadsheet, but a big cloud of dots floating in space. What PCA does is it fits a shape, an ellipsoid, which is kind of like a stretched out basketball, around that cloud. The longest axes of that ellipsoid point in the most important directions of your data. Okay, that's enough theory. Let's see how this works in the real world. We're going to look at a real PCA plot from a super interesting NASA experiment. And yes, it involves mice that went to space. And here it is. Take a look. This plot shows the results from that NASA study. Remember, every single dot you see here represents one mouse. The red dots are the ground control mice, the ones who stayed on Earth. And the blue dots? Those are the spaceflight mice. They're the astronauts. So how in the world do we read this thing? Well, the axes, you see they're labeled PC1 and PC2. These aren't genes anymore. They're the new summary variables that PCA created. Now, the most important rule is that PC1, the horizontal axis, always captures the most variation in the entire dataset. It's the big one. And you can see that dots that are close together are similar, and the two groups, red and blue, are pretty clearly separated, especially along that main PC1 axis. So how much variation are we talking about? A whopping 54%. Think about that. Over half of all the genetic differences across all 60 of these mice can be explained by this one single new dimension that PCA figured out. So what's the story here? What is the single biggest factor separating these mice? It's simply whether or not they went to space. Out of all the possible patterns in those 900 data points, PCA instantly pulled out the most important one, the experimental group. All right, so for just a second, let's peek behind the curtain at the math that makes this all happen. We're just going to touch on two terms, eigenvectors and eigenvalues, but I promise we'll keep it super simple. Think of it this way. The eigenvector is the direction of the pattern. It's the angle of that main line cutting through our data cloud. And the eigenvalue, that's the strength of the pattern. It tells us how much of the data's variation is explained by that line. So a big eigenvalue means its eigenvector is pointing in a really, really important direction. PCA just finds these pairs and ranks them for us, giving us PC1, PC2, and so on. And that brings us right back to our little analogy. The eigenvalues, they're just the principal directions of that data cloud. In other words, they're the main axes of that ellipsoid we imagined fitting around all our data points. See? It all connects. So let's bring it all home. Why does this one tool matter so much? It's all about going from overwhelming, complex data to insights that are actually clear and useful. So if you walk away with anything today, here are the big takeaways. PCA helps us see the patterns hidden deep inside complex data. It does this by reducing tons of variables down into just a few principal components. PC1 is always the most important one. It captures the most variance. And because of this, it's become an absolutely foundational tool in just about every field, from biology to finance to modern machine learning. At the end of the day, PCA is really a storytelling tool. It helps us find the hidden narrative in our data, to find the signal in all the noise. And that leaves us with a pretty exciting question, doesn't it? In a world where we're creating more data than ever before in human history, what other amazing stories are just sitting there waiting to be told?